Hello, my name is Tatiana Goncharov, and I am a scientist at Immunology Discovery Department at Genentech. I would like to share with you our finding on XIP role in TNF-induced and RIP1-mediated tests. I will start with introducing you to TNF signaling at molecular level. Briefly, binding of TNF to its receptor will result in protein aggregation and formation of receptor signaling complex, where RIP1 gets ubiquitinated and promotes further signal transduction that will result in an FKB and MAP kinase activation. When an FKB is negatively affected, or with time, RIP1 moves to cytoplasm, where it engages caspase-8 that will lead to activation of caspase-3 and 7 that will result in apoptosis. But if caspase-8 activation is compromised, RIP1 can assume active conformation that leads to autophosphorylation that enables interaction with next RIP kinase 3 that also will autophosphorylate and then phosphorylate pseudokinase MLKL, leading to necroptosis. Now, activation of RIP1 also can be due to activation of toll-like receptor 4 in response to LPS, or due to tissue damage that result in membrane permeabilization and release of DUMPs or other inflammatory factors. Here at Genentech, we develop highly selective RIP1 kinase inhibitor GNI684 with favorable properties. As a mouse model, we are using SIRS, where TNF injection will cause systemic inflammatory responses. In presented study, mice were treated with necroptotic stimuli, TNF and caspase inhibitor ZVAD, in the absence or presence of different amount of GNI684. Graph on the left depicts body temperature at 2 and 4 hours after TNF dosing. Graph on the right depicts percent inhibition by indicated doses of GNI684. In SIRS model, RIP1 inhibitor efficiently blocks hypothermia in dose-dependent manner. Within TNF signaling pathway, there is number of identified mutation in the patients with inflammatory diseases, and all of them impinge on RIP1 activation during cell death. For example, mutations in XIP lead to early onset IBD. While working with XIP deficient bone marrow-derived macrophages, we notice increased cell death responses to LPS-mediated necroptosis, here caused by LPS and caspase inhibitor emricasan, and even to LPS alone. In SIRS model, in comparison to wild-type, XIP deficient mice responded to LPS necroptosis stimuli with strong hypothermia. And in a model of GAL and LPS liver damage, we found higher levels of liver enzymes in XIP knockout mice. We also examined TNF-mediated and FKB and MAP kinase signaling pathways in bone marrow-derived macrophages, and we did not notice any difference due to genotype. But at the same time, we found that XIP deficiency enhanced cell death in response to TNF-mediated necroptosis in bone marrow-derived macrophages and intestinal organoids. And XIP deficient bone marrow-derived macrophages released more interleukin-1 beta and IL-6 under TNF or TNF-ZVAD stimulation. In SIRS model, loss of XIP synthesized mice to TNF-induced hypothermia and lethality. Increased cytokine release detected in serum and resulted in more damages detected in small intestine. Similarly, XIP-deficient mice were more sensitive than TNF mice to TNF-ZVAD necroptotic stimuli in vivo. Recently, we characterized XAP-specific small molecule antagonists and used XAP-BR2 domain selective binders in NOD2 signaling. Important to note that small molecules such as XB2 and 54 did not change the level of XAP or CAP1 protein in cells. While using XB2 and 54 molecule in vivo, we were able to block NOD2-mediated cytokine release in dose-dependent manner in mice. Consistent with our genetic deletion of XAP, dosing of XAP small molecule in wild-type mice resulted in similar effect in vivo: decreased survival, increased hypothermia, elevated cytokine levels, and more intestinal damage. At the same time, we confirmed that XAP antagonists did not alter the levels of XAP or CAP1 protein in small intestine. XB2 and 54 compound also enhance TNF-ZWAT-induced hypothermia in wild-type mice. Recently, we also worked with Tryptokinase inhibitors, testing them in NOD2 signaling. Using GSK 583 compound, we have shown that Tryptokinase inhibitor blocks RIP2 XAP binding, RIP2 ubiquitination and autophosphorylation, inhibits NF-kappa B and MAP kinase signaling in response to NOD2 stimulation. While using RIP2 inhibitor in vivo, we were able to block NOD2-mediated cytokine release in dose-dependent manner in mice. Using bone marrow-derived macrophages and intestinal organoids, we investigated involvement of RIP1 kinase activation in increased sensitivity of XAP-deficient cells. And as you can see, RIP1 kinase inhibitor G684 was very efficient in inhibition of TNF and TNF-ZWAT-mediated cell death in those experiments. Also, we show RIP1 inhibitor efficiency by Western blot analysis of signaling events in bone marrow-derived macrophages. Please note that inhibition of related kinase RIP2 does not have significant effect in these experiments. In Sears model, RIP1 inhibition restores animal survival, blocks hypothermia, and prevents tissue damages with loss of goblet cells. We also follow cytokine release during Sears at 2-hour time point and found elevated levels of CXCL1 and CCL4 in XAP-deficient mice that was significantly reduced by RIP1 inhibitor, but not by RIP2 inhibitor. Please note that the lack of RIP2 efficiency was not due to absence of RIP2 in intestines. And at 2-hour time point, we did not detect activation of proteins that are markers of cell death. So, we repeat Sears study with TNF, with takedown at 3 hours with noticeable hypothermia in XAP-deficient mice at that time. Again, we detected elevated levels of interleukin-6 and CCL4 in XAP-deficient mice that was significantly reduced by RIP1 inhibitor. Dosing of RIP1 inhibitor prevented tissue damages with loss of goblet cells in intestines. Bearing in mind elevated levels of CCL4, we examined spleens of experimental animals and found that RIP1 inhibitor blocked granulocyte depletion from spleens of XAP knockout mice after TNF injection. At the same time, we found that in the livers of XAP-deficient mice, RIP1 inhibitor shifted granulocyte levels closer to those levels detected in wild-type treated animals, with similar effect on interleukin-6 and CCL3 levels of cytokines detected in liver lysates. Importantly, that after TNF injection, Gene expression in small intestine of experimental animals were very comparable between both genotypes and was not altered by RIP1 or RIP2 inhibition. In addition, we follow cell death activation markers in the liver tissue of experimental mice and detected TNF-induced RIP1 phosphorylation and caspase-8 activation that were efficiently blocked by RIP1 inhibitor. At the same time, we did not observe any genotype differences in caspase-1 or 11 activation, in the processing of IL-1 beta or gasdermin D. And again, RIP2 inhibitor failed to inhibit activation of cell death markers. I hope that our findings convinced you that in addition to caspase inhibition, XIP plays a role in regulating of RIP1-mediated death. In gut inflammation, we observed RIP1 activation that through engagement of RIP3 and caspase-8 can cause inflammatory cell death and tissue damage and contribute to Crohn's disease and ulcerative colitis. We believe that RIP1 kinase inhibitors can block those processes. Patients with TNF-mediated and specifically RIP1 inhibition-sensitive mutations present an opportunity to implement personalized health care for inflammatory diseases. In the end, I would like to acknowledge and thank people that contributed to this work. And thank you for your attention.